So I'm really happy to introduce Professor Pratyush Tiwary. He has very interesting career, so we'll see how it's going to be because he completed his PhD at Caltech in material science. And I think he's really, his background is physics. And then he moved more into biochemistry, so it's very interesting change. And that his postdoc ETH in Zurich in Europe, and then he went back to the United States to Columbia University. Now he's a professor in the Department of Chemistry and Biochemistry at the University of Maryland. And this is very interesting talk because in this era of deep learning, when people, confuse structure prediction and structure folding, I think quite often we miss that RNAs are very dynamic structures. So it's really interesting to see and appreciate the dynamic nature of RNA and to see how we actually can combine deep learning with thermodynamic information and kind of appreciate dynamic nature of RNA molecules. So it should be fun.
Okay, so I will get started. Hopefully you guys can see my screen and hear me.
Yeah.
All good? Okay, thank you, Magnuson. Thank you, Nivedita and Rachel, for giving me the opportunity to speak here. It's super exciting. I put the result that I like most from today's presentation in the intro of this presentation itself. So you can take a look at this. RNAnneal is a method which aims to predict tertiary structure ensembles from a first principles basis. It's an AI method, but which is not a language model, and we will learn all about it. And sometimes it feels like if you're trying to compare against AI methods in RNA area, then it's easy pickings. It's like taking candy from a kid. But still, it's quite important to do that because all these methods are getting a lot of hype. And, hopefully we can convince you that physics still has, the combination of physics and AI still has a lot of potential to do important things. I'm going to start my timer here. Okay, so as it was mentioned, I'm located in the University of Maryland in College Park. I'm at last count, I am in 7 different departments and 2 different universities. So I'm a polymath. I like to do different things. And statistical physics is the core of whatever we do in my group. I also, together with my student Lucas and friend Nadia, we have co-founded a company that is commercializing this RNAnneal technology. And I will tell you a little bit about that also. So if I run out of time, I want to make sure that I thank all the people who have done the work on this. So my group is made up of students from different backgrounds, and today's work is especially due to the RNA team in my group, which is, of course, a lot of that is Lucas, who is now going to be the CEO of this company, Emergent, and a postdoc Yunrui Q, who is starting his own lab at University of Notre Dame. And I'm sure within a few years, he will be giving a talk in this series because Notre Dame has a lot of RNA work going on. And Anjali, she is this fantastic PhD student who joined my group and is pushing a lot of these methods. And others have also contributed to the RNA work in different ways. And these are my financial sources of support for the group, and these are my conflict of interest. And you mentioned in the beginning about my background. Well, it goes even more than that. And since this is a diverse audience, I want to tell you a little bit about where I come from. So I come originally from India, from northern India, just south of Nepal. I did my undergrad there, not even in material science, but in something called metallurgical engineering. Then I came to Caltech for my PhD in material science. I hopped over, went to Switzerland for a postdoc. I worked with Michele Parinello. That's why I really started getting into biological systems. I came to Columbia University to do my second postdoc, and I've been at University of Maryland. Soon I will be finishing my 9th year here. So, and it's since I started, 2017 is around when not many people were doing AI. Back then, if you wrote that you're in a proposal that you're going to use AI when you submitted your proposal to the NIH, you might have been considered novel due to the use of AI. And how times have changed. Now we are living in really strange times. And I think of these 3 as really epitomizing the times that we are living in. And the first one on the left is something called Moltbook. This has become quite famous in the last few months. This is a social network for AI agents where agents are talking to each other. Humans are welcome if they are interested. And yeah, I mean, it's as crazy as it sounds. The one in the middle, I think, is a joke, but not really. It's called rentahuman.ai. where robots or agents can ask humans to go and do something. So this is the opposite of what we have normally thought of computers, right? Normally we think of a human goes and asks a computer to do something, but this is a computer asking a human to do something. And before you say this is too futuristic, I think forms of this are already existing in society. A friend of mine here does a part-time job where they go to local grocery stores or Home Depot and things like that, and they take photographs of the different aisles to see which aisles are empty, where do they need to buy more things. And when they take these photographs, I'm pretty sure it's not a human asking them to go to which particular shop to take the photograph. The instructions are most likely coming from an AI agent of sorts. So we are already living in an era where AI is instructing humans to do things. And the one on the rightmost, I think, is the most bizarre. It's even more bizarre than rentahuman.ai. I've been working on StatMech and free energy methods for 20 years now. And to think that free energy methods will now be commercialized and put up on the side of a freeway in California, that just blows my mind. In this case, this is Boltz 2, which I'm sure many of you have played with. So these are strange times, and I would like to argue that the strange times really started with molecular structure prediction. AlphaFold winning the Nobel Prize a few years ago was the moment, AlphaFold together with, of course, David Baker's lab, and was the moment that legitimized AI. Until then, it was still, maybe it's powerful, maybe it's not, who knows. But that was the moment where, and it's not just chemistry Nobel Prize, it was also a physics Nobel Prize that went that year. And this, to me, was the moment of legitimacy, where the world was forced to acknowledge it as something very, very real. Since then, a lot of things have happened. The other moment of legitimacy is, of course, ChatGPT in the last couple of years, which has been a big breakthrough. But coming close to our area, we are continuing to see huge hype, success, whatever you want to call it. These days we see on a routine basis scientific work being published in the newspaper or in white papers without any peer review of sorts before they are published anywhere else. So, and there are lots and lots of examples, and we are continuing to be featured in the highest venues. So it all started with AI, all this hype that we are seeing. But this does not mean that all problems are solved. A lot of problems are still far beyond traditional AI. The first problem that my group is very much interested in is that with AlphaFold, we predicted, well, for well-structured proteins, we are able to predict the single most structure that is going to form. But proteins have an ensemble of structures, and often it is about predicting these alternate structures, which you can do with things like MSA subsampling, but that's not solving the conformational diversity problem. You also want to be able to predict the relative thermodynamic weights of these structures. And that is a hard problem, which we have made only partial success in solving. The second grand challenge is structured proteins are just tip of the iceberg. RNA and intrinsically disordered proteins, which I think follow a lot of similarities, are barely solved. Structure prediction for these things, especially predicting the ensemble of structures, is extremely hard. And the third grand challenge, which RNA really typifies, is that the ensemble itself, so it's hard enough to predict the ensemble, but now I'm arguing that you cannot just predict one ensemble, you have to also predict how does the ensemble respond as a function to changes in environment. The classic example, which I find quite interesting, is the RNA thermometer in the West Nile virus. When the mosquito carries it, the RNA is in a certain form where it cannot replicate and grow. When the mosquito goes and bites a bird or a human, the RNA enters a slightly different temperature, the energy landscape arguably changes, and a new conformation becomes more stable. And then this is lethal. The RNA can keep growing, it can replicate, and it's game over. So if you're just going to do this paradigm that all we need to do is to predict one structure very accurately, just falls apart. So how do we develop AI methods to solve these grand challenges? AlphaFold and these other methods and the boom in AI methods for biology that we are witnessing right now is probably because of the PDB, right? Without PDB, nothing would have happened. So if we want to come up with AI methods that can really transfer across conditions, well, one naive approach would be to come up with copies of the PDB across conditions, right? And we know RNA structures are few, but what I'm proposing is even harder. You want to get lots of structures at lots of temperatures, lots of pH, lots of chemical potential. This is this is absurd. This is not going to work. We could waste all of the money that exists in the world, and we will not have enough structures to do this, build these type of models. Furthermore, there is no guarantee that these models will extrapolate to new conditions that you did not study in this. And as I will show you slightly, environment has coupling with features. You cannot just treat it as some sort of a label in AI models that works. So how do we solve this problem? And I think, I mean, we are witnessing in AI for biology what I like to think of as the Phil Anderson moment of our generations. Of our generation. So those of you who don't know, who might not know Phil Anderson, he was this very famous physicist in 1970s. He's accredited with coining the term condensed matter physics. He won the Nobel Prize around then. And he wrote a beautiful essay that I really recommend reading called More is Different in Science magazine back then. And Phil was sort of controversial, especially by particle physicists, because he went to the Congress in Washington, D.C., and said, you should not spend all of United States science budget on building bigger and bigger colliders. Just building bigger colliders so we can collect and see what happens to physical systems if you just dig down deeper is not what it's not sufficient to answer all of physics. It's important. He was not saying we should not have that, but he said this is not sufficient. He said a lot of physics is emergent. It cannot be understood through just reducing it to small parts. You have to study systems longer, and every system has its own story. And I think we are witnessing a similar moment in AI, where the trend seems to become just get more data. All you need is data, data, data. And only if we had more data, AI will solve all our biology problems. And I think this is a dangerous approach. So as a statistical physicist, the way I look at the problem is, and what gives me the hubris to go look at different types of systems in the same one under the same roof is the simple fact that most things are made up of atoms interacting through some force field. And as we are seeing progress in force field development, one fact remains that you could have two carbon atoms talking to each other. You don't use different force fields depending on whether the carbon atoms are located in an organic material or an inorganic material, in a protein or an RNA. The underlying force field, the underlying potential energy is same, irrespective of what is the system you are simulating. And it's complex. It's got lots of local minima. It's got lots of transition states. So the interesting thing is the same potential energy, depending on what atoms do you have existing, if you simulate, if you solve, if you study the system long enough, it leads to completely different emergent behavior. This is a kinase on the bottom left, this is a crystal, and this over here is, I think, some sort of riboswitch, and you get completely different structures. So what my group tries to do is to come up with methods that combine statistical physics with artificial intelligence in a way that we can develop simulation methods that are remarkably robust and they work for these different classes of methods. I'm I'm committed to working not just on one class of methods, which means that sometimes I won't have half the depth of RNA knowledge that many people on this call have. But I think as a method developer, it's important to tackle different systems. Otherwise, our methods tend to be overfit, and that's a danger. And I will focus on the last two challenges today. I will show you primarily the RNAnneal method, which allows us to extrapolate. Firstly, it allows us to get tertiary ensembles of RNA, tertiary structure ensembles of RNA. And secondly, I will show you some work, how can we get ensembles that extrapolate across conditions. And the central idea in a lot of the work going on in my group is this needle in a haystack approach. We take AI and bioinformatics methods as sort of a hypothesis or a haystack generator. So we treat these as the chat GPT of structural biology. You have a sequence, you have no clue what's going to happen. You throw all AI methods on top of it. You throw AlphaFold, you throw Chai, Boltz, whatever you have, and even Rosetta or older bioinformatics methods, whatever you have, and you generate a massive haystack of possible structures. And then you use statistical physics plus molecular dynamics to find a needle from this haystack. So if your haystack does not have the needle to begin with, which means that none of these methods were able to get you close to the true structure, then our approaches won't work. But often it turns out that there is a lot of knowledge that has gone into these methods. Community has already done a lot of work. So there is always hope in getting the needle out of this haystack. And just brute force molecular dynamics is not going to work on this. You could, because there's just so many members in this haystack, and 10 nanosecond or 50 nanosecond of molecular dynamics is not going to tell you much. So what we have to do is to integrate statistical physics with molecular dynamics and run some form of enhanced molecular dynamics. So I will show you how this idea works. And the way we got to RNA is, before we get into RNAnneal, I have to tell you a little bit about thermodynamic maps, which is this, which really led to the RNA journey in my group. We published thermodynamic maps first in 2022. So we were one of the first groups to use diffusion models for molecular simulations. We started on this in 2020. And then if you really want to read thermodynamic maps, this 2024 paper, which has thermodynamic maps in the title, is the best paper to read about it. So I will, and thermodynamic maps is the core of RNAnneal. So I want to take some effort and explain this to you, how it works. So the motivation is quite simple. When we try to model some system in physics, we have, of course, we know the true force field is very complicated, but perhaps let's start by thinking about the simplest description of any system. What is the force through which two atoms might be talking to each other? So one of the simplest, well, the simplest description in physics is an ideal gas. Nothing talks to each other. Let's not go that far. The second simplest description in physics is a simple harmonic potential. Atoms talk to each other, but through a harmonic spring. What that means is, as you pull the spring, they like to come back, and F is equal to minus kx, Hooke's law. So if we have a simple harmonic potential where the energy then looks as half x squared, it's just an approximation to give you intuition. So let's say that we have a biological system where we have studied the distribution of distances at some temperature T1, and then we go to some other temperature T2. So if T1 is smaller than T2, then at T1, you will have a distribution which is tighter. At T2, it is a bit broader. The mean will stay the same. That is the beauty of a harmonic potential. So what does this mean? This means that if you have collected, if you know the second moment of this distribution at two temperatures, if you know the second moment at temperature T1 and the second moment, which is the variance at temperature T2, then, since this is a Gaussian probability distribution on a harmonic potential, you can fit the second moment and you can get the second moment at any temperature. Furthermore, since this is a Gaussian potential, this is a Gaussian probability distribution, we know that the first moment, the third moment, the fifth moment are going to be 0. And knowing the second moment gives you information about all higher order moments. That's the point here. So in statistical mechanics, or in statistics, people call this the moment generating function. The idea is that if you have collected enough moments of a probability distribution, you can construct the full probability distribution. And the point I am making here is that if the probability distribution is harmonic, it's a Gaussian probability distribution, then just knowing the second potential is enough to construct it at any temperature. So this is very interesting. This means that if you knew the variance at one temperature, you now can go and construct the full probability distribution at any temperature. And if you can construct the full probability distribution at any temperature, you should be able to draw samples at any temperature. Okay, so this is simple, really, really simple idea. But of course, nothing is harmonic in real life. Everything is complicated. God knows what is the underlying potential, but the same intuition can still be used. We no longer have the luxury of analytical relations now between the moments and temperature, but the same idea exists that if you knew the first moment, the second moment, the third moment at some temperature, let's say not just one temperature, but 2 or 3 temperatures, then maybe you have a way of learning the moments at other temperatures. And if you are able to learn all the moments at all temperatures, then suddenly you have a way of generating samples at any temperature of interest. So you don't need to fit the energy landscape of the RNA at 2 temperatures. All you need to do is to see where does it sit on an average, what is the fluctuation from this average, possibly the third moment. If you have this information at 2 temperatures, then suddenly you can go and interpolate and extrapolate across temperatures. This is the intuition. In practice, what we find is that the first moment and second moment tend to do a remarkably good job, better than we thought they should. So we rarely go to third moment. Well, we never go to third moment. And I will give you some more intuition into this, how it looks. So let's say you have done simulations or experiment on your biological system at 4 different temperatures: a very cold temperature, a cool temperature, a hot temperature, and a very, very hot temperature. You take all these things. I see Giovanni Bussi is on the call. So Giovanni is one of the experts on replica exchange type simulations. So this could be coming from a replica exchange simulation also, where the exchange probability is very weak. If you had very high exchange, then of course, you don't need to do any of this business. You can already go and do stuff at any temperature. So what we do is to take this information collected from 4 different temperatures, we throw it into a magic pot. I really like to read Asterix comics, so I always think of the magician in Asterix comics called Getafix. Imagine his magic vessel. So you throw it into this magic vessel in this pot. And now what we want to do is to train a generative AI model which takes this information and generates samples at any temperature. So even though I said replica exchange, if you had very good exchange, would work, even replica exchange won't allow you to generate samples at other temperatures. It's not tricky. But what we want to do is to come up with a generative AI method which allows you to, first of all, improve the sampling quality at the cold temperature, because the hope is that at very hot temperature, you are going to sample lots and lots of metastable states. And that information can now be used to improve the sampling quality at any temperature. So this is what we are going to do. And the reason why this is possible is due to this motivation over here, that if we have sampled enough means and variances at 1 or 2 or 3 temperatures of interest, it should be possible to construct information at other temperatures of interest. And my PhD student, Suimin, she is now in 4th year and is looking for postdocs if anyone is interested. She, together with Ruyu Wang, another postdoc in the group, was able to generalize thermodynamic maps to not just extrapolate across temperature, but even pressure and chemical potential. This is super important now because, for example, pH is a form of chemical potential. So suddenly you have AI models which can extrapolate not just across temperature, but even pressure or chemical potential. So I will show you the most classic example in statistical physics. This is called the Ising model. So this is a bunch of spins which can point up or down. At very low temperature, if you start, so let's look at these 2 movies. The top one is at low temperature, the bottom one is at high temperature. Both start with the same randomized configuration. At low temperature, you get a very ordered phase. One phase disappears and you are left with either all spins pointing up or all spins pointing down. In absence of an external magnetic field, the system is degenerate. So you might end up with all up or all down. That degeneracy is there, but the point is the modulus of the magnetization will be 1. At high temperature, the system will stay disordered and the modulus of the magnetization will be 0. So the question is, what is the temperature, the critical temperature at which this change happens? Furthermore, what happens to the heat capacity of the system as you go through this critical temperature? And you might say, what is all of this got? I thought I came to an RNA lecture. What does all of this got to do with RNA? Quite a lot. So just hang in with me. So this Ising model is a system which people have used to, you can model everything with an Ising model. You can model magnets, you can model human brains, you can model voter behavior. This could be yellow could be right-wing voter, blue could be left-wing voter, and every neighbor is talking to his neighbor, and let's see how voting tendencies appear. So people use Ising model for everything from science to social science. But it's really challenging, and at least 2 Nobel Prizes have gone for solving the Ising model using accurate methods. And especially, we know in this system that there are long-range correlations close to the critical temperature. So the heat capacity of the system actually shows a power law behavior. As you get close to the critical temperature, it diverges. So in this follow-up, in this thermodynamics maps paper in PNAS 2024, we showed, Lucas and a rotation student in my group, Kinjal, they showed that what we can do with thermodynamic maps is to collect information at only 2 temperatures. One temperature is far in the fully ordered phase, and one temperature is far in the fully disordered phase. And these temperatures could lie anywhere. The only thing that matters is they should not be on one side of the critical temperature. They should be on either side. So they could be symmetric, asymmetric, whatever. So these are the red stars over here. So you have data only from 2 temperature, But you train thermodynamic maps on this, and you can get the magnetization curve at any temperature of interest with very, very high accuracy relative to a brute force, much longer Monte Carlo. Not just that, you can get the spike in the heat capacity and the critical exponent within remarkable accuracy compared to the Monte Carlo simulation. I mean, you might say, "Oh, this is very high error," but I mean, this is a second order phase transition, and you're getting the spike, even though you never saw that happening. So this was very encouraging to us, and we thought, well, like most statistical physicists, if we can do Ising model, we can do everything. And it was a bit of hubris, but then we saw that, yeah, we can make progress in RNA. Oh, yeah, and this was the thing that I mentioned done by Suimin in this paper, which is on arXiv, where we generalized it to extrapolate across other things also, not just temperature. So yeah, we were not the only ones who thought that Ising models can be used to model RNA structure. Parisi, who got the Nobel Prize in Physics a few years ago, back in the day, he had a paper on this where he came up with a very accurate map between spin glass and an RNA secondary structure. So this was very inspiring to us. But then this paper has like 10 citations or something like that. No one cares about it because what Parisi did was, of course, to set up an exact model, and you do all sorts of exact things to solve it. We are not as smart as Parisi, and we are in the era of AI. So we are going to use AI, and we are not thinking only about secondary structure. We are thinking about tertiary structure also. So the jump from Ising model to RNA is not that hard. It's basically a complex Hamiltonian. We don't want to learn the terms of the Hamiltonian, or rather we have access to the Hamiltonian. It's very hard to simulate for long enough. And so we will start very slowly with small RNAs, okay? So a tetraloop, GCAA, and HIV tar hairpin. Of course, HIV tar hairpin, as you guys probably know better than me, has, thanks due to Al-Hashemi and others, there is a lot of different structures that have been collected. So what my postdoc, Hyunrui, who is now starting his own lab in Notre Dame, did was to use all of my supercomputing resources, nearly, and did like millisecond long MD simulations of these systems, as you can see here. So we have very, very as accurate of a benchmark as we can have. We want to compare apples to apples, right? So that's why we did MD, which is very long, because we never, I mean, force fields for RNA is another elephant in the room. We don't know what's quite going on there. So we wanted to be comparing the same force field from thermodynamic maps with 1 millisecond long or fraction of a millisecond long MD simulation. So, and yeah, I mean, and I'm conflating a couple of papers here. Not all papers are using the same method, but the idea is the same. So already in this PNAS paper, we had shown that for tetraloop and for HIV tar, we can start recovering structures which are deposited in the PDB without ever having seen them. And we can even get, just like the Ising model, we can get the melting temperature. There is this in good agreement with experiments or with very long, in this case, Anton simulations. You might notice that the experimental melting curve is much sharper than what we are getting out of thermodynamic maps. There are a couple of questions here. I think, how exactly do you count fraction unfolding? This is a fudge factor. You can change this and your curve will become sharper. But the point is that the temperature at which melting happens is quite accurate. And what Yunrui did is to show that we can not just get the melting curve, we can now start getting the temperature-dependent free energy landscape. So on the left, you have it for a tetraloop. On the right, you have for HIV hairpin. And you can see how rugged it is. It is anything but a funnel. So for the tetraloop at 300 Kelvin, you can count the number of states. It's at least 10 states. For the HIV tar, there are many more. And I don't think we have published it yet, but the preprint should be out soon, where you will see that the dynamical ensemble reported by Al-Hashimi, we can actually recover most of those structures and even the NMR factors in the Szabo sense. But at high temperature, the states just disappear. You start to see one entropic blob for both systems. And one of the other things we can do on this is to come up with, we can build a Markov state model on this, and we can actually study the kinetics for RNA folding and interconversion between different states. And it's complex. It's not a two-state picture, of course. There is a whole zoo of transitions that's going on. What is quite fascinating is that at low temperature, and here I'm showing you for the tetraloop, at low temperature, even though you have such a complex landscape, At low temperature, you can fit the folding time with, at high temperature, you can fit the folding time even for this one with a single exponential kinetics. At low temperature, even though you have so many different states that exist, you can still fold it with a double exponential kinetics. And that's kind of fascinating for two reasons. First of all, at high temperature, it indeed, at 400 Kelvin, it's following a very nice single exponential. And that at 300 Kelvin, you don't need to go to more terms than this. A bi-exponential really does a very good job of capturing this. So Dave Thirumulai, who used to be in Maryland, exactly in the building where I'm speaking to you today, had done beautiful theory on this back in the 90s and later, and still works on it, where he had come up with theories that explain these things of how you are either directly following into the native state, or you go to misfolded state and then you go into native state. And what we are doing is to really fit to Dave's theories and show you how we can recover those kinetic constants. So, and there is a ton of other papers that we also have on this. Before I forget to mention, I learned a lot of my RNA business from my collaborator, Jesh Niklat at National Cancer Institute. And with him, we have many other papers on this topic. But we started getting confidence into the business of developing RNA structures. We even took part in the CASP competition 2 years ago. It was a disaster. I mean, we failed terribly. We did. I would say we did not even fail. It was like could not have done worse. It was horrible, but we did not give up. We kept improving the method, and we are very proud of it now. So, and the method really does thermodynamic maps, and that's why I explained all of it to you. But to generate the initial structure for thermodynamic maps, it uses a whole bunch of secondary structure prediction methods that exist out there. Our patent application is now public, so you can go and read all of the details of it. Even the bioarchive is out, so everything is now disclosed in that. You can see what are the whole bunch of different secondary structure prediction methods we use. How do we generate our haystack? But the point is, once we have the haystack, we do thermodynamic maps, and we get our ensemble of structures, not just one structure, but the whole ensemble at the tertiary structure level, together with their populations. Furthermore, we can change these populations as a function of temperature. This is fully unsupervised. The method I'm going to show you next, it takes around 12 hours to get the whole ensemble for one RNA on a GPU. And I think it's not AlphaFold speed. It will probably never be AlphaFold speed, but it's high accuracy from what we can see. And one of the classic examples is the SARS-CoV RNA. You know, this is one of those where, of course, we know it was not there in the AlphaFold tree training database. So if you just run AlphaFold tree on it, you get a wrong structure compared to the cryo-EM. You guys know cryo-EM more than me. I think I would say, though, even this is an underestimate. It probably costs more than this. And our number, we have now actually pushed it to become even cheaper because it's just one GPU running for 12 hours. So it's cheaper than this. And you can see that we never trained our model on the experimentally known structure for this one. So it had never seen it, yet it does a good job of getting it. What's even more interesting, that if you see our preprint, we talk about that not only are we getting this structure, we are also able to quantify the uncertainty on a per nucleotide basis, which correlates quite nicely with with that is what is measured in the cryo-EM experiments. So that's just one structure. We can go and look at a bigger database of structures. These are ribo switches. When we submitted this preprint, we were not making progress that much with pseudoknots. Which is we undersold ourselves. Since then, we have also made a lot of progress with pseudoknots. So now we don't have any issues with pseudoknots also. So for these 16 different ribo switches, I'm now showing you, and this plot itself is now doing better. I don't have the updated plot here today, but it still carries the important message. So one of the main messages here is RMSD on its own is not the best metric to be looking at RNA structure quality. This is something that is well known to many organizers of the RNA competitions, RNA puzzles, and many others, that you need to look at alternate metrics. You need to look at either ERMSD, or even better, you need to look at a 2D space. You need to look at RMSD together with the network fidelity. If you go to very low RMSD from the reference structure, then sure, there is a correlation. Very low RMSD corresponds to very high INF. But as you start getting to the 7 to 8 RMSD difference, which is where a lot of structure prediction method lies, the same 7 RMSD deviation does not mean all structures are same. There could be a huge range of INF values for the same 7 RMSD deviation. So what we are doing here is we get to very high accuracy. So I think Lucas, who is also on the call, can correct me, but we are following standards reported in one of the RNA puzzles paper. So if we get RMSD within 5 angstrom and INF more than 0.85, we would call that high accuracy. We get that to at least 5 out of 16 structures. And moderate accuracy, which is this range, we get to 6 of 16. And we got to low accuracy in 5 by 16, but with some changes, we are doing even better here also. And furthermore, we are not just getting one structure, we are getting a whole ensemble of structures, and together with their probabilities, which I think is a novelty of what we are doing over here. And this plot is unpublished, but I'm really proud of this one, and I would love to hear what you guys think. So let's take a moment to go through this carefully. These are 23 RNAs, which are different from the ribose switches that I showed you before. So now 16 plus 23, we are looking total at basically 40 RNAs. So these are 23 RNAs. All of them are smaller than 100 nucleotides. So our RNAnneal currently has its sweet spot up to 100 nucleotides. It does okay up to 200 nucleotides. If you go beyond 200 nucleotides, it starts to gasp, it gives up. It's not very good there, but we are getting there. We are continuously improving in that area also. But up till 200, we are very happy with it. So here, let's look at this one. I'm showing you a bunch of RNAs for which in the left figure, I have measured the INF for RNAnneal, for AlphaFold 3, for Chi and Boltz. And in the right one, for all these 4 methods, I have measured the RMSD deviation. So of course, INF higher is better, RMSD lower is better. Let's start with RMSD. If you look at the overall data set, RNAnneal is giving accuracy which is comparable to AlphaFold 3. At this point, you will be like, well, then what do you do? You're just getting close to AlphaFold 3. But if you look at the INF, we are looking, like I said, you have to look at both things together. For INF, we are doing much better than AlphaFold 3. That's for the overall. What is most interesting if you look at the synthetic RNA? By synthetic, I do not mean funky nucleotides. I mean just sequences that are new. The sequences are synthetic. Here, the differences really stand out in both metrics. In RMSD, we are, I would say, doing reasonably better than these other methods. And even in INF, we are doing other methods. And you can look at other classes also. And we have, this is something we are working on actively. So we have even more data points every day we have more data points for this, especially as we work with collaborators, which is something I will talk about briefly. We keep improving, we keep finding that the method is more accurate than what we thought earlier it is. And you can do a bunch of things with RNAnneal. First of all, you can predict high resolution structure models. Like I showed you, these are some of the RNAs we have looked at. And we are definitely not against experiments. We have a collaboration going on with Danny Incarnato, and every time I speak to Danny, I learn so much. Danny and Jay, they have been showing us how to get shape data or chemical probing data and other things, and how to use those as priors in our model. We will use whatever information you have from different experiments and improve our method or be consistent with that. And all of these methods are really haystack generation methods. You can use all the structures that you're getting out of these AI methods to generate the haystack for RNAnneal and then rank them. So we are not in competition with these methods. We are per construction going to be superior to them because we can rank them. And finally, if you're thinking about doing free energy perturbation on RNA structures, this is a nice paper my friends at Schrödinger published. Free energy perturbation for proteins already is difficult. If you have large conformations, changes in the protein. For RNA, well, the name of the game is conformational changes. So this quantity delta G conformation is important. And that's exactly what RNAnneal is providing to you by looking at the free energy, by giving you the probabilities of different conformations. And so I think this is my final slide. I want to leave some time for questions and answers. So the preprint is out. You can go and read it. It's with one of the editors at one of these journals, you can guess. And although if it comes back, we have already added so many more RNA since we submitted the first preprint first time, that we can improve it even more. And we have commercial interest in RNAnneal because we have a spin-off, Emergent with an E in the end, where the E stands for emergent engineering. And we are thinking of RNAnneal as the, let's say, the CUDA for the AI revolution. It's a method which can be used for a lot of different RNA problems. So we are actively fundraising for it. But at the same time, we are making it completely free for academics. We are working heavily with a student, with Lucas and with a student Gokul, who is now in my group, to make it available on cloud so everyone can use it. And you won't be able to see the code, but you will still be able to run it. And if you have a sequence where you don't want to share the sequence with us, we can set it up even in such a way that we don't see your sequence. You can run it directly. And for we have some money to run it for academics. Even we will pay for your GPU costs. You don't even have to pay that to us. Of course, we don't have that much money, so we have some money for that. And depending on targeted problems, we can do that for you. But otherwise, we have a way. If you're willing to pay for the GPU, that's even better. We can get it going. And I think that is my final slide, and I will be happy for any questions.
Okay, thank you. Thank you once again. Very interesting talk. I can start with my own question. It seems that as you explained, like you have to start with relatively good structures from these deep learning methods. So I have two questions, like what would happen if you don't really have in the first pool of structures something that is really similar to native? Is the method is kind of able to, take what is as best as possible and kind of get into native states?
Yeah, so there is some amount of deviation there. So if it is, uh, let's take Malat1 as an example, which forms a triple helix. And if none of your secondary structure methods or bioinformatics methods are getting you close to a triple helix at all, then this won't go and discover the triple helix for you. But the point is that if in this haystack, which can be quite big, this can be a million structures, if one of them ever visits a triple helix, then we are good to go. Then it will work. So yeah, it has to be somewhere close to it. Does not have to be exactly close to it. It has to get somewhere close to it, and then we can do the remaining job.
So, and the second part is more to kind of explore this idea. Do you see any way how you could integrate thermodynamics deeper into like deeper into AlphaFold like systems? Are you have any ideas? Do you have any ideas about it?
I think the whole thing will need to be changed the way currently AlphaFold is working. So one of the things, why were we able to so we also use a diffusion model, which AlphaFold 3 also uses, so that is common. And I don't think it's too hard to do it, but they will have to revisit their whole framework of how they are tackling the problem. It can be done, but it's not trivial. So what we are instead thinking about is to just use AlphaFold type methods uh, uh, as where did it go? As the haystack generation part of the process.
Okay, yeah, yeah, we have a few questions from Miro. I can read them. Are these vanilla unbiased MD simulation or replica exchange simulations?
No, these are vanilla
Yeah.
unbiased empty simulation. To push it to run only 12 hours on a GPU,
So-
you can see all of the details in the preprint. We have disclosed
Yeah,
everything.
sorry. The
Yeah.
question was about the like 300 microsecond runs for the tar RNA and
Oh,
the
for the tar, that's
yeah. I
that
was just
is
curious.
okay. No. So
Like all this.
that is explicit, everything explicit, and not replica exchange, but Markov state model calculation,
Okay, but
not replica exchange,
the individual runs are just unbiased. Like they're
just
not
unbiased
exchanged.
one.
Okay.
No
I
replica.
see. I see.
No,
Okay. So
because
you
if
ran
you
it
go
at
to
two
replica
temperatures
exchange
and stuff.
with 40,000 atoms, you have the exchange probability will start going down.
Yeah. Thank you.
No problem.
Miro, you can go again if you want
Oh,
to ask
yeah.
your
So
question.
yeah, I had another question. I had another question about um the predictions you showed for the COVID RNA. I was just curious if they were like so like you're predicting an ensemble, but here you're saying, oh, I'm very close or I'm doing better than AlphaFold
Yeah,
or
yeah, yeah.
something. I'm just curious how you then select the structure from
Yeah,
your haystack.
that's a very good point. So for these ribo switches, I am showing you the candidate needles from the haystack, right? We have a bunch of
Yeah.
different structures that we are getting. For the coronavirus, I think, Lucas, you are on the call. Do you want to answer this question?
Yeah, I can.
Yeah, go ahead.
So yeah, for the coronavirus, we do predict this conformational ensemble, right? Some discrete set of structures. We get that set of structures from the haystack. We use this other method that has been developed in Pratyush's group called SPIB, State Predictive Information Bottleneck, to basically assign states to the thousands of structures that are in the haystack. And then we choose the best representative among the states calculated by the RNAnneal score. And if you want to, you can see that in the preprint. In this case, we predicted this particular coronavirus RNA. We predicted, I believe, 8 different representative conformations. Most of them were consistent with the secondary structure that you're seeing here. You see how it's kind of T-shaped. The other predictions that we made kind of had the top of the T wobbling on this almost like hinge point where the two helices meet.
Right.
This one was the second ranked one that we're showing out of the 8. And the reason that we chose that second ranked one was that visually, it was the closest to this cryo-EM.
I see. So yeah, okay. So you're still like, yeah, that makes sense. Thank you.
Yeah, no problem.
Thanks, Lucas. More questions?
Nivedita, do you want to say it? I can also read it. So Nivedita is interested more into details of MD simulation. So OPC and T4PD were recently reported to destabilize RNA structures by Jiří group. Could you please comment on the choice of water and force field model for simulation?
Yeah, absolutely. So I kind of conflated 2 stories over here. If you look at these stories where we are getting the accurate landscape and everything is super well resolved and converged free energies and networks like this, here I think, and I don't know if Yunrui, are you on the call?
Oh, yes.
What force field did you use here?
I used the most recent initial research Amber-based force field.
Yeah.
Together with a TIP4PD. And of course, as mentioned by one of the audience, like this force field may destabilize the RNA structures. But I think this is the most updated force field we can use. And of course, there are also other shortcomings with other force field.
Yeah. Yeah. Thanks, Yunrui. So one point I want to make is, and I think I made it very clear in the beginning itself that this is primarily a sampling talk. So what we are trying to do is to come up with methods where we can switch out the force field. If you want to run it with a better force field, you can use our methods to do that. In fact, in RNAnneal, if you go and look at the preprint here, you will find that forget about the Shaw or which version of Amber you are using. Here, we really brought it down to coarse-grained models, implicit solvent water, which speeds up the process. And turns out for getting to this level of accuracy, we are making a lot of progress without all atom resolution force fields. If you want to dig deep, if you have a RNA where you really want to understand what's going on better, you really want to understand what are the ions doing, then you can take these ensembles that you get out of RNAnneal and use it to seed much more accurate molecular dynamics with newer force fields, even polarizable force fields like Alex McCarroll, who is my colleague at Baltimore. You can use his force fields even. So yeah. So basically, to answer that question, we for the RNAnneal platform, we try to be as fast as we can. Once we get to our ensemble, depending on the needs of the end user, if you're an academic group, you want to study one RNA very carefully, now you have the opportunity to seed more MD simulations, even seed more replica exchange MD simulations starting from RNAnneal ensemble with any accuracy force field.
There is another question about force field.
Mhm.
Charges in force fields usually don't change. How does it affect your simulation as RNA is a highly charged system?
Could you repeat the beginning part?
Charges in force fields usually
Oh.
don't change. How does it
Yeah.
affect your simulation as RNA is a highly charged system?
Yeah, that's a wonderful question. In fact, if I look at the Amber force field that we used for some of them, and Stefano Piana at D. Shaw kindly shared the files with us. The charges were funky, like sodium had a charge of 1.33 and things like that. We have used what we got in the force field. We have not played around much with altering the the force field charges. And again, you're most welcome to try things like that, because now we have, given any set of charges, we have a sampling that you can trust. We ourselves have not explored that direction of what happens with changes in the charge, but would love to see the community try those type of questions.
Okay, Karim, you can go ahead. I'm not sure what was the order, but go ahead and ask your question. Yeah.
Thank you for your great talk. It was so inspiring.
Thank you.
I'm wondering, have you tried to, bring ML or AI in first to a step into the structure prediction and molecular dynamics simulation to reduce the cost or make it more efficient?
We could, but what we found for RNA is, if I go back, where do I go back for that? I don't think I have a slide really talking about the details of what we do to generate the initial haystack. But what we found here is that the bioinformatics, roughly calling it bioinformatics type methods that have come up for RNA over the decades, are super fast and are really accurate. And this is something which has come out in CASP competition after CASP competition. Purely AI approaches are being left in the dust by methods that are using bioinformatics. So we didn't see the point. I mean, what is the point? Because bioinformatics is always going to be faster than ML. So we did not even try it. But and I don't know if that is going to improve. So a second part of my group does similar problems in proteins. In fact, I spoke in CASP special interest group last year in the AlphaFold Rave method. There it makes a lot of difference. There, of course, AI methods have sped up all of this. MSA subsampling in AlphaFold does a very good job. And also to answer your question, this is sort of what I was referring here, is we could speed up the process by I wouldn't call it speed up, because the quality of structures coming out of one of these methods is not exactly reliable. So I don't know if I would trust it. It's a long answer to your short question.
Thank you.
We have one more. Since you generate a sample, how good of a single structure is representative to the ensemble? And the effect of ions can be injected into the framework.
Yeah, that's a really, really good question. So in fact, even if you go back to this one, if you zoom in over here, so here what we have done is to project the free energy along 2 PCA coordinates. Notice the NMR structures don't lie smack in the middle of the free energy minima. They are somewhere in the basin, but they are not in the minima. So this already shows you that the force fields are not exactly reproducing the perfect NMR structure. So we are getting the ensemble. We are getting something which is close enough to the experimental structure, which sorts of explains something. If you look at the figure I showed over here, or actually, let's go to this one. I mean, on an average, our RMSDs don't just drop down to extremely low values. Well, none of the other methods do also, but I won't be surprised if the RMSDs coming out of our method don't get to 2 angstrom, 3 angstrom accuracy. What they do a good job of is landing you close enough to the experimental structure. And I think this difference between our RMSD at the very final stage versus experiments will improve as we see parallel developments in RNA force fields with accurate sampling methods.
Okay, we have one more. Could you elaborate a bit on how one performs MD simulations to generate haystacks regarding the selection of conformational space similar to the Ising model needed to have sampling sandwich TC?
Yeah, so what I'm going to do at this point is, since I'm getting so many questions about this, I'm just going to open the preprint instead of asking you to go read the preprint and go back to sharing my screen, because I removed that slide for some reason. I don't know why I removed it. And I think, Sorry about that. So, by the way, thank you very much for all these nice questions. It's super, super fun. So if you go over here, there you go. So this is really all the credit goes to Lucas and Anjali and Yunrui. So we start with Eterna4, Linear4, and other things to generate a lot of secondary structures. It's 10,000 here, but sometimes we go to even more. Then we do FAR2 and clustering on top of this. Then we get a bunch of tertiary structures. And this is where the first molecular dynamics comes in. There is no molecular dynamics until here. This molecular dynamics is also not even all atom. This is coarse grained. And then we generate a candidate ensemble of structures. And even though some of these methods are mentioned over here, we are continuously adding more and more methods to generate a better haystack. So that's the first step. And then you can see here, so we use the Amber force field with implicit solvent model. And it's when we put things into the model, the first thing is, how do you characterize an RNA? How does a model see an RNA? Is it all atom distances? Is it all atom coordinates? No, that's a terrible idea. Even though AlphaFold 3 did it, we don't have that much GPU. You will have to worry about equivariance and all that business. So what we do is to generate a bunch of G vectors and all these other things. Then we do dimensionality reduction on top of it. And there is a bunch of nuance that goes in over here. And what we can do is to generate different models. We do, roughly speaking, for example, you can do PCA with 2 PCA, PCA with 3 PCA, PCA with 4 PCA, and you don't know which PCA is correct. You don't need to know which PCA is correct. What we can do is to take all of those and throw them into the thermodynamic maps landscape. And that's when we start to get the ensemble of structures together with the probability. So that's the whole framework. And yeah, please see this. Yeah, go ahead.
So could I ask a very quick question about
Please.
FAR? So I mean, you could spend all your CPU on sampling FAR structures. I'm just kind of curious whether you like have a limit in the number of Monte Carlo cycles you do, how much minimization you do.
Lucas?
Yes, so I've used FAR2 so much in the course of this project. We do, I think in the results reported in this paper, we do like 10,000 Monte Carlo cycles over 10 rounds for each candidate structure plus minimization. But then lately we found that maybe you don't need to go to 10,000 rounds, especially if your RNA is on the smaller side. So lately I've been using 5,000 cycles. And then sometimes minimization is beneficial, sometimes it's not. That's also up to your discretion maybe, but I do tend to use minimization.
Yeah, I find it produces pretty wacky stuff without it. Okay, thank you.
Yeah.
Okay, so maybe Emily, the last question, and then we should close.
Hi, this is Emily Tai. I'm from NCI and NIH, so
Hi,
we
Emily.
are neighbors. Yeah,
Yes,
so
we
this
are.
is fantastic talk. Yeah.
Thank you.
So two things I'm curious. One is that I saw you have your methods. RNAnneal is very good at RNA protein prediction. The INF score is very high. So this part is very good. So I want to know, one thing is that how in your next slide, I think, or the previous slide, you said use RNAnneal to model tertiary structures compatible with experimental constraints, because even we here at NCI, so we have collaborators who are study like a bacteria, small RNA, and HFQ and other small proteins interactions. So they really want to know about the interactions between the protein and RNA. So that's why I'm very excited to see your performance is very high. But can you elaborate a little bit more how to use your method to model the tertiary structures? They have a lot of experimental data, not the structure, but it's more like a biochemical, like a different conditions. So how to use that? Thank you.
Yeah, that's wonderful. So first of all, please send me an email and Lucas and I will talk to you. Okay. So I really look forward to that. NCI, like you said, you are my neighbor. We can come and talk even in person. So what we can basically do is that somewhere in this pipeline, and I forget where exactly, probably somewhere around here, whatever information you provide us can be added as constraints. So the tertiary structures that come out will be consistent with that constraint. Now you mentioned RNA proteins. One of the things we would love to do is to really generalize this to generate the full RNA protein structural ensemble at the same time. Of course, that's a really hard problem. All this progress that has happened in the RNA area is sort of orthogonal to some level to the progress that has happened in the protein area. We don't know how to do that yet, but already we can start to give you insight into where the protein might come and sit. Yeah, so we would take any constraints you give us and put them somewhere over here. We have a fairly automated way of doing that. And so let's talk more.
Oh, great. Thank you.
No problem.
Okay, with that, I think we can close for today. Thank you again for a very interesting talk and all the discussions. And I think we see each other in 2 weeks.
Okay, take care.
See you.
Bye.
And, have fun.